Hello everyone. So today we are here with new video on anticoagulation mechanism. So we have already discussed that how coagulation takes place. So we are going to discuss how anticoagulation mechanism takes place in our body. As we know that if coagulation is taking place at the site of injury, this is very important to limit the coagulation to the site of injury only, right? And when there is wound healing after that, we have to stop the coagulation also. So what are the mechanisms that takes place in our body for this anticoagulation? First of all, let's know about the limiting factors which are going to limit the coagulation process. First of all is dilution. What do you mean by dilution? When the blood starts flowing through the site of the injury or the site that is healed and where the coagulation factors have been accumulated, they will wash out these coagulation factors and will take these coagulation factors towards the liver. Okay, so blood flow will take the coagulation factors which were causing coagulation, platelet aggregation, will take them to liver, and there in liver they will be degraded. Second limiting factor is requirement of negatively charged phospholipids. Uh, as we discussed in that coagulation, for the platelet aggregation, there is a requirement for this negatively charged phospholipids for platelet aggregation and activation. And if these negatively charged phospholipids get used up or exhausted, or we don't have these negatively charged phospholipids, it will become a limiting factor towards coagulation. Third is the fibrinolysis. That in coagulation we know that fibrin is formed, right? Fibrinogen is converted to fibrin and fibrin gets polymerized, right? So if there is fibrinolysis, means these fibrin threads are getting lyased, it will lead to fibrinolysis, and it's also a limiting factor towards coagulation. It will also stop or hinder the coagulation. Let's have a look on the fibrinolytic pathway. So if this question is asked in your exam about the fibrinolytic pathway or simply the anticoagulation mechanism, you can simply write this pathway only. First of all, what we need to know that how this fibrin is getting broken down. So it's this fibrin is getting broken down with the help of plasmin, right? This plasmin is helping to break down the fibrin threads and interferes in its polymerization. As we know that fibrin threads gets polymerized and they form a clot, then there is clot retraction, etc, etc. So this plasmin will interfere in this polymerization and it will lead to the increase in number of the fibrin split products. The fibrin fibers or threads will be splitted, so there will be increased number of fibrin split products. What are these fibrin split products? These are fibrin derived D dimers. Okay, so where does this plasmin came from, right? Where does this plasmin come from? What is the source of the plasmin? So plasmin comes from this plasminogen. And where does this plasminogen comes from? It is freely available in our bloodstream. Okay, so plasminogen is in its inactive form and it gets converted to plasmin in its active form with the help of this TPA. What is this TPA? TPA is tissue plasminogen activator. Tissue plasminogen activator. Okay, and this tissue plasminogen activator is available through the intact endothelium, right? So intact endothelium, which is not exposed uh, endothelium. So it's released from this intact endothelium and converting this plasminogen, which is inactive, into the plasmin. Now this plasmin, we know that plasmin is going to interfere in the polymerization and it's going to break the fibrin threads. So how this plasmin is getting controlled? We need to control, we need to keep everything in like balance, right? So we need to control the plasmin activity also. So plasmin activity is controlled by this alpha 2 plasmin inhibitors. Okay, endothelium is very important over here in the anticoagulation mechanism as well as coagulation mechanism. As we discussed in coagulation, that endothelium get exposed, right? There's a tissue injury, endothelium get exposed, endothelium gets removed and subendothelial substances get exposed. What comes in the subendothelial space like von Willebrand factor, collagen, and they stimulate the aggregation of the platelets. These all we discussed in the coagulation cascade and coagulation mechanism. So, what is the role of endothelium in the process of anticoagulation? Let's have a look on endothelium. So, what is endothelium doing? First of all, it is secreting, it is providing a platelet inhibitory effect. What is the platelet inhibitory effect? We know that platelets are very important for coagulation. They aggregate, they form a clot, and then they entrap the blood cells, and then fibrin gets accumulated and they form a clot, right? So what is endothelium doing? Endothelium is shielding, it is shielding the platelets from getting activated, from helping the platelets to not come in contact with the von Willebrand factor. We know that there is endothelium layer, and beneath this there is subendothelial substances like von Willebrand factor and collagen, and here platelets are here. So this is providing a kind of shield. This platelet now can't get in contact with this von Willebrand factor and collagen, right? It can't come in contact with them, so it is providing a shielding effect. Second thing, it is secreting these substances PGI2, NO, and ADPase. What is PGI2? PGI2 is a prostacyclin, which is a type of prostaglandin formed from the arachidonic acid metabolites that will study in biochemistry very well. So it secretes prostacyclin. It secretes NO. What is NO? NO is basically nitric oxide simply. There is endothelial nitric oxide synthase which secretes this NO, forms this NO. Then there is ADPase. ADPase, what will it do? It will help in the lysis or digestion of the ADP. So what they are doing collectively, they are collectively inhibiting the aggregation and activation of platelets. Okay, second effect what endothelium does is anticoagulant effect. It secretes certain other factors also like thrombomodulin, endothelial protein C receptor, heparin like molecules. We know that heparin, right, anticoagulant, heparin like molecules, tissue factor pathway inhibitors. So here in this diagram we can understand it very well. So here you can see this is an endothelium that I have made. This is endothelium, endothelial cells. So here you can see that endothelium releases what? Heparin like molecules. These heparin like molecules are going to bind with the antithrombin third. Antithrombin third in turn binds with the thrombin and inactivates the thrombin. Along with thrombin, it also inhibits the factor 9th A and 10th A. 10th A we know Stewart-Power factor. These factors are playing role in the coagulation cascade, right? So they are inhibiting these factors and thereby inhibiting the coagulation itself. Second, tissue factor pathway inhibitors, which will inhibit the release of the tissue factors, right? And also inactivates the 7th A. 7th A tissue factor 7A complexes basically. We knew that in the extrinsic pathway of coagulation cascade, this tissue factor binds with the 7th coagulation factor, right? So this complex is inhibited or inactivated with the help of this tissue factor pathway inhibitor. Third is thrombomodulin. Thrombomodulin is binding with the thrombin, and then it's kind of activated by this activated protein C, which is a kind of a vitamin K dependent protease. It activates this protein C. What is protein C? It is a vitamin K dependent protease. Vitamin K dependent protease, which requires protein S to act, and it inactivates the factor 5th A and 8th A. Okay, so what other it is doing, what other things, what other effects endothelium is showing? It also releases this tissue plasminogen activator, as we discussed earlier that it shows fibrinolytic effect. It will convert the plasminogen into plasmin, and plasmin will break down the fibrin threads and also interfere in its polymerization. So that's it for the anticoagulation pathway. Very easy, right? Okay, thank you.